What if I told you that right now, as you're watching this, trillions of cells inside your body are doing exactly what you're about to see in this video? Today, we're looking at one of the most iconic videos in all of cell biology, a time-lapse of a human cell dividing, pulled straight from Gray's Anatomy. And trust me, once you understand what those glowing red and green colors actually mean, you will never look at mitosis the same way again. Welcome back to Gray's Gyaan, where we take the dense, complex topic of Gray's Anatomy and break it down so clearly, you'll never forget it. Let's get into it. So before anything else, let's decode what we're actually seeing in this video. There are two colors. That's it. But each color is telling you something completely different. The red signal, that's Histone H2B, labeled with a fluorescent protein called mCherry. Now, Histone H2B is a protein that DNA literally wraps around to form nucleosomes, the basic packaging unit of your chromosomes. So whenever you see red in this video, you are looking at chromosomes. You are looking at DNA. The green signal, that's Alpha-Tubulin, labeled with EGFP, enhanced green fluorescent protein. Alpha-Tubulin polymerizes to form microtubules, and during cell division, these microtubules form what we call the mitotic spindle, the molecular machine that physically pulls chromosomes apart. So in one sentence, red is your chromosomes. Green is the machine that moves them. Now, why HeLa cells? Why these particular human cells? That story is extraordinary. The year is 1951. A 31-year-old woman named Henrietta Lacks walks into Johns Hopkins Hospital in Baltimore, complaining of abnormal bleeding. She's diagnosed with cervical cancer. During her treatment, a sample of her cancer cells is taken, without her knowledge, without her consent. Those cells are sent to a researcher named Dr. George Guy. And here's where something almost miraculous happens. Every other human cell that scientists had ever tried to grow in a lab died within days, but Henrietta's cancer cells? They kept growing, and growing, and growing. They were, for the first time in history, immortal human cells. They were called HeLa cells, HeLa, from the first two letters of her first and last names. Henrietta Lacks passed away just months later, at just 31 years old. But her cells? They are still alive today, in laboratories across the entire world. Since 1951, HeLa cells have been cited in over 110,000 scientific publications. They helped develop the polio vaccine, the HPV vaccine. They were critical to COVID-19 vaccine research. And the very video we're analyzing today was filmed using HeLa cells. Normal human cells die after around 40 to 60 divisions. HeLa cells divide indefinitely. It is one of the most important, and most ethically complex, stories in the history of medicine. And we owe Henrietta Lacks an enormous debt of gratitude. Now let's talk about how scientists actually made chromosomes and microtubules glow, without killing the cell. This is where it gets really clever. Technique number 1, mCherry. mCherry is a red fluorescent protein that was originally derived from a sea coral. Scientists took the gene that codes for mCherry and fused it directly to the gene for Histone H2B. So now, every time the cell makes H2B protein, it automatically makes it with a red glow attached. No dye injection, no chemicals, the cell itself is doing the labeling. Technique number 2, EGFP. EGFP stands for Enhanced Green Fluorescent Protein, and it won the Nobel Prize in Chemistry in 2008. Scientists fused the EGFP gene to Alpha-Tubulin. So every microtubule that forms inside the cell glows green, automatically. Technique number 3, time-lapse microscopy. A confocal fluorescence microscope photographed the cell once every 3 minutes, over several hours. When you play those frames back at normal video speed, hours of biology compress into seconds. And here's the most elegant part. Both of these fluorescent genes were permanently integrated into the HeLa cell's DNA. So every daughter cell inherits the glow. The experiment runs itself. Alright. Now we understand the tools. Let's walk through what actually happens, phase by phase, and connected directly to what you see in red and green. Interphase. This is the cell at rest, before division begins. The red signal is a soft, diffuse glow throughout the nucleus, because DNA is loosely packaged as chromatin. The green signal shows a web of microtubules spread throughout the cytoplasm, the interphase cytoskeleton. Prophase. Watch the red signal carefully here. It starts to condense. The diffuse glow sharpens into distinct, thread-like chromosomes. This is chromatin packing down into its tight, compact form. Meanwhile, the green signal starts reorganizing. The centrioles duplicate and begin migrating to opposite poles, and the spindle starts to assemble. Prometaphase. This is when things get dramatic. The nuclear envelope, the membrane surrounding the nucleus, breaks down. And now the green spindle fibers can reach in and grab the chromosomes. Each chromosome has a protein structure called the kinetochore, and the spindle microtubules attach directly to it. Metaphase. Look at the red signal. All the chromosomes have now lined up perfectly at the metaphase plate, an imaginary line right in the center of the cell. This is the cell doing a quality check, making sure every single chromosome is correctly attached to spindle fibers from both poles. It is extraordinary precision. Anaphase, and then it happens. A protein called separase cuts the connection holding sister chromatids together, and suddenly, the red signal splits. Chromosomes are pulled, rapidly, to opposite poles of the cell. The green spindle simultaneously elongates, pushing the poles further apart. This is the moment of no return. Telophase. The chromosomes arrive at the poles. Watch the red signal now. It begins to decondense. Two separate blobs of red appear. Those are the two new nuclei forming. The nuclear envelope reforms around each set of chromosomes. The green spindle begins to break down. And then, the final act. Cytokinesis. The cytoplasm itself has to physically divide. In animal cells like HeLa, this happens through a structure called the cleavage furrow, a ring of actin and myosin filaments that forms around the middle of the cell and contracts, like pulling a drawstring on a bag, tighter and tighter, until the cell pinches in two. What you see in the video is the cell elongating, a narrow bridge appearing between the two halves, called the midbody, and then, separation. One cell becomes two, each one genetically identical. Each one carrying a full copy of the chromosomes. That is mitosis. Let's take a moment to appreciate just how incredible the green signal really is. The mitotic spindle, made of microtubules, is actually 3 different types of fibers, each doing a different job. Kinetochore microtubules attach directly to chromosomes at the kinetochore. During anaphase, they physically shorten, literally depolymerizing from the chromosome end. And that shortening is what pulls the chromosomes to the poles. The force of life, at the molecular scale. Interpolar microtubules run from one pole, past the equator, and overlap with fibers from the opposite pole. Motor proteins grab onto these overlapping zones and walk along them, pushing the poles apart and elongating the spindle. Astral microtubules radiate outward toward the cell membrane. They position the spindle correctly in the center of the cell, and they help determine exactly where the cleavage furrow will form. And here's a bonus fact that will blow your mind: microtubules are not static. They are constantly growing and shrinking in a process called dynamic instability, catastrophically collapsing and then regrowth again, all in seconds. In the video, that flickering green glow? That's thousands of tubulin molecules adding and removing themselves, live, in real time. So why does any of this matter clinically? 1. Cancer drug testing. HeLa cells with these exact fluorescent labels are used in cancer research labs worldwide to test anti-cancer drugs. Drugs like Taxol, paclitaxel, work by stabilizing microtubules, preventing the spindle from functioning. In the video, you'd see the green spindle freeze, and the cell die. This is how researchers screen new chemotherapy candidates. 2. Aneuploidy and disease. If the red chromosomes don't separate equally, if one daughter cell gets an extra chromosome and one gets too few, that's called aneuploidy. It's the mechanism behind Down syndrome, Turner syndrome, Klinefelter syndrome, and it's present in the vast majority of cancer cells. 3. Anti-mitotic chemotherapy. Vincristine, colchicine, paclitaxel, these drugs all target microtubules. Understanding spindle dynamics directly shapes how we treat cancer. 4. Nobel Prize science. The EGFP technology used in this very video won the Nobel Prize in Chemistry in 2008, awarded to Osamu Shimomura, Martin Chalfie, and Roger Chun. It fundamentally changed our ability to watch biology happen in living cells. Alright, before we finish, let's lock in the 6 things you absolutely must take away from this video. Number 1. Red is H2B mCherry. It labels chromosomes. Watch it condense, align at the equator, then split and decondense. Number 2. Green is Alpha-Tubulin EGFP. It labels the mitotic spindle. It assembles, separates chromosomes, then disappears. Number 3. Mitosis has 5 phases: prophase, prometaphase, metaphase, anaphase, telophase, followed by cytokinesis. Number 4. HeLa cells are immortal cancer cells from Henrietta Lacks. They are the most widely used human cell line in the history of biomedical research. Number 5. Fluorescent protein tagging, EGFP and mCherry, allows us to see living molecular processes in real time, without killing the cell. Number 6. Errors in mitosis lead to aneuploidy, which drives cancer and genetic disease. This is not just biology. This is the foundation of clinical medicine. That's it for today. From a caption in Gray's Anatomy, to the full story of fluorescent microscopy, immortal cells, and the molecular machinery of life. If this helped you see mitosis differently, give it a like, share it with a fellow med students, and subscribe so you don't miss our next video. With this video, today the first chapter, Basic Structure and Function of Cells, is completed. After this, we will begin the second chapter, which is integrating cells into tissues.